Hello everyone, a very warm welcome to Ashish Biology. Today we will discuss about Mendel and his experiments, and this will be the first part from the chapter Principle of Inheritance and Variation. Before starting this video, let me introduce myself. I am Ashish Kumar Das, a student of MSc Animal Biology, University of Hyderabad. If you are new to this channel, please subscribe the channel and press the bell icon for getting more updates. Now here we go. So first of all, what will we learn today? We will see what are Mendel experiment and the plant used as research material for Mendel's, that is by Mendel. What is an allele? What is a gene? What is law of dominance? What is law of segregation? What are monohybrid and dihybrid crosses? Law of independent assortment, and many more. We are going to discuss in this topic. So before going into the topic, let us recapitulate some of the important terms that we have studied in our previous classes, that is in our junior classes, and it will be very much helpful for us to know, that is to reconnect the earlier concept with the new one that we are going to study. So first of all, genetics. So genetics is the branch of science that deals with inheritance and variation of character. So inheritance and variation, these are two new terms that is coming here, that is we are encountering here. So first of all, inheritance, that is the process of transmission of character from the generation to one generation to next generation. So most of the organism, whether they may be sexually reproducing or asexually reproducing, will reproduce their newer generation or offspring, and what happens, they will transmit their character from them to the next generation. Then variation is the character differences among the individual of a species. So character differences means the various type of differences, maybe in face, maybe in height, maybe in weight, etc. So just imagine, no two siblings are exactly alike, except in case of twins. No two siblings are exactly alike. That's why what happens, the siblings are the closest relative and they are not alike. So you can just imagine between two cousin and two far distance, that is persons. So the character differences is there, and this is called variation. Then gene. So gene is the unit of inheritance, and it codes for a mRNA or protein. So this concept will be much more clear when we will see the next topic, or when we will go further. Then allele. Allele is the slightly different or alternate form of a gene. So chromosome contain genes, and genes is having allele. So allele is slightly different or alternate form of a gene. Now come to phenotype. So phenotype means the observable or external characteristics of an organism or plant. For example, if you are seeing a plant, that is any plant or a person, you may see its height, color, etc. Then in case of flower, you can see the color of the flower, then the position of flower, etc. So these are the external characteristics you can see. Then come to genotype. Genotype is the genetic constitution, that is the chromosome, combination, number of chromosome present. So these are the genotype. So genetic constitution is the genotype, which is invisible, that we can't see genotype. We only can determine this by making various scientific investigation or crosses, you can say. Now some more terms, that is homozygote and heterozygote. So homozygote or pure breeding means individual organisms in which the member of a pair of allele or a character are similar. So what happens, the individual in which the member of a pair of allele, so each allele is having each gene is having two allele, so the member of the pair of allele are similar here. So this is homozygote or pure breeding. Then come to heterozygote or hybrid. So here the members of a pair of allele are dissimilar or different, so this will be heterozygote. Then dominant factor or allele. So dominant allele is the allele that express itself in a hybrid, and the character encoded by the dominant allele is called dominant character. So in a hybrid condition, that is in a heterozygote condition, some allele express themselves and they will be dominant allele. Then in the recessive allele are the allele which express itself only in homozygote condition. So you need two copies of the allele to show the recessive character. So the allele which are required in two number, that is two allele is required for the showing that phenotypic expression, they will be called recessive allele, and the character encoded by recessive allele is the recessive character. Then monohybrid cross between two individual considering the inheritance of a contrasting pair of a single character. So here you are considering single character, that is contrasting pair of a single character. So this is dealing with only one character. We will see about details, that is character and trait. Then dihybrid cross made between individual of a species considering the inheritance of contrasting pair of two characters. So in case of dihybrid cross, you have to consider two character. Now Mendel experiment material. So Mendel conducted several experiments on garden pea, that is Pisum sativum, from that is for 7 years from 1856 to 1873. So you can just imagine his dedication towards his experiments. So he selected pea plants because of two reasons, that is many varieties are available with observable alternate forms of a trait or character. So a huge number of varieties of pea plant is available, that is the scientific name Pisum sativum, and the alternate forms, that is the observable alternate forms, that they are clearly distinguishable. Even you can simply see them by naked and you can imagine the differences. Then peas are generally self-pollinated or self-fertilized. So self-pollinated means it will undergo self-pollination. That means it is having both male sex organ, that is stigma, and female sex organ carpel in the same plant. That's why they can undergo self-pollination. And even if you are removing the anther or stigma, they will be cross-pollinated. So they can be easily cross-pollinated and the self-pollination can be prevented easily. Now you can see the character. So character are the, that is the, you can say the properties by virtue of which you can, that is by virtue of which you can distinguish some plants. So for example, by the virtue of seed shape, you can see different type of seeds are there. Seed color, you can see different type of seed color. So character are the, that is the attributes of a plant or organism by virtue of which you can distinguish two plant or more plants or animals. So seed shape, dominant means round, that will be expressed in both, that is your homozygote and heterozygote condition, and wrinkled is recessive. Then seed color, yellow is dominant, green is recessive. Flower color, violet is dominant, white is recessive. Pod shape, full, that is full is the dominant and constricted is the recessive. So here full pod and constricted, the full is also called as inflated pod. So for example, you can see this is a full pod, and if you are seeing like this, just imagine this will be a constricted pod. So full pod and constricted pod, I hope the concept is clear. Then pod color, green color is dominant, yellow color is recessive. Flower position, axial is dominant and terminal is recessive. So axial is dominant means you can see the position, that is if this is the mid axis, the flowers are arising from the sides, that's why this is axial. And here you can see this is the axis, the flowers are arising from the, that is the at the end of the axis, that's why this is terminal flower. Then the stem height tall, you can see the height is tall here, so this is dominant, and the dwarf is recessive. So this set of characters, that is round, yellow, violet, full, all of these are dominant, and the others, that are the wrinkled, green, white, that are the recessive. Now Mendel's approach, what did he has done? We will see Mendel's approach. So his methodology included investigation of inheritance, that's why he was mainly working on the in determining the inheritance pattern in the pea plant, and he used the mathematical logic and the statistical analysis. So this value, that is sorry, this thing is very new at that time. So that is the scientist working in the biological field rarely using mathematical logic and statistical analysis. So you can see this is a new innovation by Mendel. Then he was having a large sampling size, that is he collected a large sampling size, the large number of plant he have collected, that's why it is giving higher credibility to the data he collected. So the credibility of data collected is that it is of a large sampling size, so it is giving more credit to the data. Then the confirmation of inference from his experiment on successive generation of his best plants proved that results are pointed to general rule of inheritance. So the various inference he are getting from his experiment, and that is he is doing for successive generation, he is not lefting after doing only one generation. So the results, the inference you can see proved that the results pointed to the general rule of inheritance. Now Mendel made sure that each of the 14 varieties, that is you have considered 7 character, and that is the 14 variety means each character is having 2 type of plant, that is 2 contrasting form. For example, if you are taking plant height, we will get tall and dwarf. So he made sure that all of them must be true breeding, and by allowing successive generation, so allowing, that is he allowed successive generation to self pollinate, and eliminate offspring that was not true breeding. So he developed true breeding. So for example, question may come how he developed true breeding varieties. So he was allowing self pollination for a number of generations, and this is ensuring true breeding, or you can say homozygote. Then he studied the inheritance of each of the 7 characters individually by conducting monohybrid crosses. So each of the 7 characters we have discussed, and he was first conducting monohybrid crosses. So monohybrid means he was considering for a single character, and then in combination by conducting dihybrid and trihybrid crosses. For example, if he is taking plant height, flower color, it will be dihybrid. If he is taking plant height, flower color and pod shape, it will be trihybrid. So more than one character he was taking 2 trihybrid or dihybrid crosses. Then he allowed each offspring to self-pollinate to produce F2 generation. So parental generation is their first filial F1 generation, then F2 generation, second filial generation. Subsequently F3 and F4 generation are also produced, and they are also raised by self-pollination. And he also conducted test crosses. So we will discuss what is test crosses. So Mendel observation. So F1 hybrid showed that one of the parental forms of the trait, so that is parental hybrid, that is after the parental you are getting first filial, that is the F1 hybrid, showed that any one of the parental forms of the trait, that is one of the, that is the parental is getting suppressed in the F1, then both the parental forms of the trait, contrasting forms of the trait, appeared without any changes in the F2 generation. So in F1 generation you are getting only one of the parental form, but in the F2 generation you are getting both the parental forms. So this is Mendel observation. Then the 2 contrasting forms of a trait doesn't show any blending, and either in F2 or that is F1 or F2 generation. So there is no blending. Blending means, for example, if you are taking a tall plant and a dwarf plant and you are getting an intermediate plant, that time you can say you are getting a blended, that is the blended result. But here what happens, there is no blending, they were recovered as such, they were having, means in the parental. Then the form of trait that appeared in the F1 hybrid is called the dominant form. So we have discussed dominant. So F1 hybrid is showing one of the parental form, and that will be dominant, and it appeared in the F2 generation about 3 times in frequency as it is alternate recessive form. So in the F2 generation ratio was 3:1 between the dominant and the recessive. So the F1 hybrid is that is the form that is expressing the, or the F1 hybrid is expressing a form and that will be dominant, and in appeared and is appearing in the F2 generation 3 times as compared to the recessive. Now Mendel inference, what the inference he derived. So Mendel made the following 3 inferences from his observation. The characters are controlled by the factors, that is the plant height or flower color, they are controlled by some factors. He named it a factor. Now we are calling this as genes and alleles. So they are stably passed down without any change from the parents to the offspring through gametes. So there are no changes when they are passing from the offspring, that is passing from the parents to the offspring in the form of gametes. The factors occurs in pair, that is the alleles. Nowadays factors are called alleles, and the alleles always occur in pairs, that is the contrasting form. Then if in a pair of dissimilar factor of a trait, one of them dominates over the other and expresses itself, while other remain hidden and is recessive. For example, if you are having 2 allele, then one allele will express itself and another will get suppressed. So the one that is remain suppressed will be the recessive allele, and it will remain hidden. So now come to monohybrid cross. So monohybrid cross means the cross having, that is it is considering the only one character. So here we are consider the character plant height. So you can see TT, that is the homozygous, small t is also homozygous. So here you are getting, that is the gametes capital T and small t, and they are mating to form capital T small t and capital T small t. So as you know, capital T codes for the tall and the small t coding for the dwarf, that is the tallness and dwarfness. So capital T will be dominant, so capital T will be tall, and here it will be tall. So only F1, that is the F1 generation, is showing only tall, but the F2 generation is showing both tall and dwarf. So both tall and dwarf is showing in the F2 generation. So you can see now in the F2 generation you are getting 3 tall plants, these 3 are tall, and only one, that is the dwarf plant. So the ratio will be 3:1. So phenotype, genotype we have considered. So genotype all are TT, so no ratio. Phenotype all are tall in the F1. In the F2 generation you can see 1 is to 2 is to 1 is the genotype ratio. So capital T 1, this is 1, 1, these are 2, capital T small and small t 1. So 1 is to 2 is to 1, and the phenotype ratio is 3 is to 1, that is tall is to dwarf. Now law of dominance. So law of dominance, so this is Mendel's law. Law of dominance states that in a pair of contrasting character, one of the inherited trait will be dominant and the other will be recessive. So first of all, contrasting pair of character. So capital T and small t, that is tall and short, are two contrasting pair of character. Why they are contrasting pair? Because you can easily look and contrast among themselves. So in the F1 generation, they are producing tall and tall. So in a pair of contrasting trait, one of the inherited trait will be dominant. So here the capital T, or you can say tallness is dominant. That's why it is that is the small t or dwarfness will be recessive. So one of the trait will be dominant, another will be recessive. So don't confuse yourself with character and trait. Here the plant height is the character and the trait is the tallness and dwarfness. So you can see the same phenotype and genotype ratio here also. So this will be the, that is, you can see here, this will be genotypic ratio. By mistake it is written phenotypic. Now law of segregation. So law of segregation, this is another Mendel's law. So law of dominance or law of segregation is derived from the monohybrid crosses. So law of segregation states that allele segregates randomly into gametes. When gametes are formed, each allele of one parent segregate randomly into gametes. So here the allele segregate randomly, that is the segregation, that is the separation of one allele is independent of other, and it is not a rule, hard and fast rule, that one gamete will get this allele, another will get this. They are segregated randomly. There is no any hard and fast rule like this. So the homologous chromosome carrying the capital T gene, that is for tallness, another is carrying small t gene, that is for dwarfness. So meiosis is there and the pairing of homologous chromosome will be there, capital T small t, and after there segregation will be there, capital T gametes and small t gametes. So half of the parents get, that is half of the parents gamete carry each allele. So here half of them carrying the capital T allele and another half will be carrying the small t allele. Now come to dihybrid cross. So we have already defined dihybrid cross means these are the cross considering the transmission of two type of characters. So here we have taken, that is the round yellow, means the flower, sorry, the seed shape and seed color. So all are dominant, round yellow is dominant, capital R, small y. After the selfing, sorry, after the cross, you are getting round yellow, that is capital R, small r. So this is heterozygote, this is the heterozygote, and these two are homozygote, or you can say true breeding. Now you can get the ratio, you can see. So here we can generate four kind of gametes, that is capital R, capital Y, small r, capital Y, capital R, small y, small r, small y. Similarly we can get here four gametes. So 4 gametes into 4, there are 16 offspring, and as we know the capital R is dominant over the small r, so yellow is dominant over the green. Similarly you can see again the yellow, then again the yellow, you can see yellow dominant over the green. Similarly round is dominant over the wrinkled seed. So here you can get the ratio 9 is to 3 is to 3 is to 1, that is round yellow is to round green, wrinkled yellow is to wrinkled green. So law of independent assortment, this is derived from the, that is the second, that is the dihybrid cross. So it states that genes of different traits can segregate independently during formation of gametes. So the genes of different traits, so here you can see we have considered two characters, that is round yellow and wrinkled green, that is the seed shape and seed color. So the transmission of the one character, that is the one character trait, for example round or wrinkled, is independent upon transmission of yellow or green. So it describes how different genes, that is the for gene for round or gene for the, that is the roundness means the seed shape and the that is the seed color yellow, is independent upon each other. So independently separate from one another when they produce reproductive cell or gametes develop. So here you can see round, that is the capital R is for, that is the roundness, and small capital Y is for the, that is, you can see here, you take like this, capital R small r is for roundness and capital Y small y is for the yellow. So the transmission of the capital R small r, that is the roundness, that is for seed shape, and the capital Y small y, that is for seed color, is independent upon each other. There is no hard and fast rule that that with capital R capital Y will come. There is nothing like that. That's why here we are getting 4 different kind of the gametes. So you can see here capital R can combine with capital Y and small y. Similarly, small r can combine with, that is your capital Y and small y. That's why we are calling this as a random and law of independent assortment. So r can independently assort with y, it can also assort with small y. Similarly, small r can assort with y and it can also assort with small y. So this describes how different genes independently separate from one another when they produce the gametes. This is the end of the video. Thank you for watching. Take care.